Hello, good afternoon everyone. My name is Suraj Vajanath Chaudhary. Today the topic of my seminar is plant tissue culture. Let's see some contents of plant tissue culture like definitions, history, types of culture like seed embryo culture, root culture, callus culture and etc. So plant tissue culture. Plant tissue culture is a technique of growing a complete plant in vitro from a part of a plant such as leaf, root, shoot, stem and etc. Plant tissue culture is mainly based on totipotency. Totipotency is the capacity of plants to regenerate a complete plant from any part of plant. Types of culture involved in plant tissue culture like seed culture, embryo culture, root culture, callus culture and etc. First, seed culture. Seed culture is a technique in which a complete plant or a seedling can be generated in vitro by taking the seed as explant. This technique is primarily used for orchid plants as the seeds of these plants do not germinate well in vivo conditions. Orchids live in a symbiotic relationship with the fungus from the moment of germination. It has been reported that orchids can be grown independent of fungus in vitro by substituting the fungus with a nutrient medium. Next point, embryo culture. Embryo culture is a technique in which isolation and development of mature or immature embryos in vitro to produce a complete plant. There are two types of embryo culture. First one is mature embryo culture and second one is immature embryo culture. Embryo culture being routinely used in crop improvement programs to obtain interspecific hybrids. Next type, root culture. Root culture is the culture of apical or lateral root tips to produce in vitro root system. Root tip culture are maintained in an agitated liquid medium with appropriate oxygen. These are mainly based with inoculation or Argobacterium rhizogenes to produce secondary metabolites. Next culture is callus culture. Callus culture is a technique through which a complete plant is formed from undifferentiated mass of cells under aseptic conditions or nutrient media. Callus culture are slow growth plant culture system in static media which enables to conduct several studies related to many aspects of plant like growth, differentiation and metabolism. Thank you.